The DNA molecule contains two strands of nucleic acids. I've drawn here two very small strands of nucleic acids. These two strands of nucleic acids are held together by hydrogen bonds, and the hydrogen bonds exist exclusively between the bases on one of the strands and the bases on the other strand. Even more specifically, the hydrogen bonding exists between the thymine nucleotide and the adenine nucleotide, or between the guanine nucleotide and the cytosine nucleotide. Those are the only combinations that can actually hydrogen bond with each other. Adenine and thymine, when they hydrogen bond together, they hydrogen bond only in two locations, which I'm drawing right here with these little dots. So those are the two spots where they connect to each other. Cytosine and guanine, when they hydrogen bond with each other, they actually have three connections, which I'm showing with these dots right here. And so this is all that's holding these two strands of nucleotides together to make DNA. Once the two strands get held together, each strand twists itself into a helix shape, and this creates what we refer to as a double helix because it's got these two helices that are just sort of intercoiled within each other. Notice that one of the DNA strands runs from 3' on the bottom to 5' on the top, while the other strand runs from 3' on the top to 5' on the bottom. So they're running in reverse direction. This helps to create a perfect pairing between the nucleotides on one strand and the nucleotides on the other. So if you are provided, for example, with one strand of DNA, so let's say that somebody says there's a strand of DNA and the nucleotides are, as I'm drawing right here, from 5' to 3', C, C, A, T, G, A, T, CT, we'll say. This is a very small strand of DNA. And if somebody asked you, could you please write the complementary strand to this strand of DNA? When they're asking for the complementary strand, they're asking you to write the other strand that's attached to this DNA. So you're going to be writing it in the complementary direction. So you'll be writing it from 3' to 5', and you'll be keeping in mind that the cytosine nucleotide can only bond to the guanine nucleotide. So if the 5' strand has a cytosine in this first position, then that means the 3' strand needs to have a guanine, one guanine for each cytosine. And then likewise, if you have an adenine, the complementary strand needs to have a thymine. Or if you have a thymine, the complementary strand needs to write an adenine. So this is a pattern that you'll just pick up pretty quickly if you haven't already mastered it. When you're writing the complementary strand, you just write the base, the nucleotide base that complements that particular nucleotide's base. And we refer to these combinations, C and G, and also A and T, we refer to them as complementary base pairs. Complementary, because they complement each other. They are bases. They are pairs of bases that complement each other in DNA.